To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.
Right, children. Now we are going to start the exercise chapter of this lesson. So as always, you all have to write the answers before me, and when I explain the answers, you can correct your answers and check whether they are correct or not. Okay? So let's start. So we are going to start with the exercise given in the textbook. Okay? Number 1. Name the parts labeled as A to D in the picture of the microscope. Look at this one, children. The picture is given. Now it's very easy because we learned how to label the parts. Okay? What is A, children? A. What is A? We'll first write A, B, C, D. A is the lens that is closest to the eye, that is known as the eyepiece. Eyepiece. B is here, you all can see if you observe carefully, coarse adjustment knob. Coarse adjustment knob. What about C? C is the stage. And what is D? Here you all can see it's pointed at the mirror. Okay. A is eyepiece, B is coarse adjustment knob, C is stage, and D is the mirror. Right? Next one. The magnification of the eyepiece was 5 times and the magnification of the objective lens was 40 times in a particular microscopic observation. What is the magnification of the microscope? What is the magnification, children? What is the formula we can use to find out the magnification? We will write that first. Magnification of the microscope. How to find out? Let's write equals magnification of the eyepiece multiplied by the magnification of the objective. Objective lens also correct. Right. So according to this, the magnification of the eyepiece was 5 times. Right. Magnification of the objective lens was 40 times. So what is the answer? 200. 200 times. Right, children? So magnification of the microscope equals the magnification of the eyepiece multiplied by the magnification of the objective. Right? Next one. Write 2 factors to be considered when a microscope is used. What are the factors to be considered? Like microscope is an expensive object. When you use it, you have to be careful. What are the things we can do to protect this object? So when you carry the microscope, you have to carry it with both the hands, right? Your familiar hand should be used to hold the arm and the other hand should be kept under the base. And you have to, when you carry it from one place to another place, you have to always take it towards yourself. Right, children? So the other thing is, after using, you have to clean the stage using a clean piece of cotton. And you always should use clean cover slips and clean slides. Okay? Those type of things we can write. So you have to write 2 factors. Wipe the stage with a clean piece of cotton after using the microscope. Use clean slides and cover slips. Right. 2 factors to be considered when a microscope is used. Wipe the stage with a clean piece of cotton after using the microscope. Use clean slides and cover slips. So we have completed the exercise given in the textbook. Now we are going to start the extra questions. We'll move on to the next. Okay, exercise number 1. Underline the most suitable answer. The resolution of the naked eye is, what is the resolution of the naked eye, children? 0.1 millimeters. 1 millimeter, 2.5 millimeters, 0.1 millimeter, 0.25 millimeters. Answer number 3. 0.1 millimeters. Which of the following instruments has the highest resolving power or highest resolution? Out of all the microorganisms that we've learned about, which one has the highest resolving power, children? Electron microscope. Compound light microscope, electron microscope, simple microscope, and the naked eye. That is electron microscope. What is used as a simple microscope? What do we use as a simple microscope, children? We use only one convex lens or magnifying glass. Okay? A convex lens, a concave lens, 2 convex lenses. We use 2 convex lenses in the compound light microscope. Okay? A concave mirror. The answer is convex lens. A convex lens. The magnification of the eyepiece was 10 times and the magnification of the objective lens was 20 times in a particular microscopic observation. What is the magnification? How to calculate the magnification? You have to multiply the magnification of the eyepiece by the magnification of the objective lens. So what is the magnification of the eyepiece? 10. What is the magnification of the objective? 20. 10 into 20, 200 times. Right? Answer number 2. Right, children? Number 5. The body tube of a compound light microscope is adjusted using, mainly to move it here and there a lot, we use the coarse adjustment knob, and to move it finally, we use the fine adjustment knob. So the answers given: the stage, coarse adjustment knob, the diaphragm, the eyepiece. Stage is used to place the specimen on. The diaphragm is used to control the amount of light received to the microscope, right? Eyepiece is it is the convex lens that is closest to the eye, right? Therefore, the answer is the coarse adjustment knob. Number 6. If the microscope is not used for a long period of time, the lenses should be removed and stored in. What is that airtight jar-like structure? That is known as the desiccator, right? A dry box, water, a desiccator, any of the above? No. You should place it where there is no water. Therefore, water cannot be the answer, right? So, dry box. If you don't have a desiccator, you can even store it in a dry box with silica gel, right? But the most correct answer is a desiccator. The microscope with 2 lenses was first made by which person made it for the first time, children? Zacharias Janssen, right? So even though the microscope made by him was not that powerful, the microscope with 2 lenses, which means the compound microscope, was first invented by Zacharias Janssen. So here Antony von Leeuwenhoek, he actually developed, and he is the first person to observe the microorganisms under the microscope made by him, right? Zacharias Janssen, Galileo Galilei, Albert Einstein. Answer is Zacharias Janssen. Even though he invented the microscope, it was not powerful. Its magnification was only 9 times. Okay. Anyway, the answer is Zacharias Janssen. In a compound light microscope, the amount of light obtained by the specimen is controlled by. Which part controls the amount of light? That is done by the diaphragm, right? The diaphragm, the mirror, the objective lens, the body tube. The diaphragm. Right, children? We'll move on to the next. Right. Exercise number 2. Write the appropriate part of the compound microscope for each description below. Now we are very familiar with this. The closest lens to the eye. There are 2 lenses in the compound light microscope. The lens that is closest to the eye is known as the eyepiece. Right? What is the other lens that is closest to the specimen? That is known as the objective. Right? The closest lens to the eye, eyepiece. Eyepiece. The closest lens to the specimen, objective lens. Objective lens. This is used to adjust the body tube to get a clear image of the specimen. What is the main knob that is used to adjust the body tube to get a clear image of the specimen? Cause adjustment knob. Cause adjustment knob. This is used to adjust the body tube finally to get a more clear image of the specimen. After adjusting with this one, to get a fine image, we use the fine adjustment knob. Fine adjustment knob. This is used to keep the slide of the specimen. Where do we place the slide on? On the stage. Right? The closest lens to the eye is the eyepiece. The closest lens to the specimen is the objective lens. This is used to adjust the body tube to get a clear image of the specimen, that is the cause adjustment knob. This is used to adjust the body tube finally to get a more clear image of the specimen, that is the fine adjustment knob. And this is used to keep the slide of the specimen, that is the stage. Right? We'll move on to the next. Right. Exercise number 3. Define what resolution is. What is the meaning of resolution or resolving power? The minimum distance there should be between 2 dots, 2 points, in order to be identified as 2 distinct points. Okay? Let's write. The minimum distance in which 2 points must be separated in order to be seen as 2 separate points. Right? The minimum distance in which 2 points must be separated in order to be seen as 2 distinct points. Right? What is the resolving power of a healthy human eye? What is the resolving power? 0.1 millimeters. Right, children? Moving to the next. Okay, exercise number 4. Define what the magnification of a microscope is. Let's write. The number of times the specimen is enlarged or magnified. specimen is magnified by the microscope. Right? Number of times the specimen is magnified by the microscope is called the magnification. Okay? Write the formula used to calculate the magnification of a compound light microscope. Very easy. Magnification of the microscope equals to, what is the answer, children? The magnification of the eyepiece into the magnification of the objective lens. The magnification of the eyepiece multiplied by the magnification of the objective lens. Right. Write the formula used to calculate the magnification of a compound light microscope. Magnification of the microscope equals the magnification of the eyepiece into the magnification of the objective lens. Right? Right. Number 5. Tabulate the differences between the simple microscope and the compound light microscope. We already did this. Therefore, this is very easy. So, this side simple microscope and this side compound light microscope. Let's write number 1. Here simple microscope, only one convex lens is used. Here 2 convex lenses are used, right? 1 convex lens present. We'll write very briefly. 1 convex lens. Here 2 convex lenses. And here the magnification and the resolution are lower, here higher. Okay? Magnification and resolution are low. Here magnification and resolution are high. Right? Here structure is simple, structure is complex. Simple structure, here complex structure. This can be used to magnify the objects, the small objects that can be seen with the naked eyes. This can be used to magnify the small objects that cannot be seen with the naked eye, right? Let's write that as well. Magnifies the small objects that can be, you can already see them, this just magnifies them, right? That can be seen with the naked eye. Here, magnifies the small object that cannot be seen, which means the microorganisms. Seen with the naked eye. Right? So simple microscope has only one convex lens. Here, two convex lenses present. Here the magnification power and the resolution power are lower than that of the compound microscope. Simple microscope structure is simple. It has only a handle, frame and the convex lens. Here the compound light microscope, it has a complex structure, right? The simple microscope magnifies the small objects that can be seen with the naked eye. Here it magnifies the small objects that cannot be seen with the naked eye. Right, children? We are moving to the next. Exercise number 6. Write 3 factors to be considered when using the compound light microscope. Now we know many things to consider. We will write. So we know that when we store this compound light microscope, you have to always store it in an upward position. Right? You should not tilt it. We will write. The microscope should be stored in an upward position. Upward position. Next one. If the lenses are not used for a long period of time, the lenses should be removed and stored in a desiccator with silica gel. Right? Let's write this one as well. If the microscope is not used for a long time, the lenses should be removed and stored in a desiccator with silica gel. We have to write 3. We'll write the third one. We should not change the lenses of lens of one microscope to another microscope. Okay? Do not change the lenses of one microscope to another. Right, you have write only 3, but there are other examples as well. So after using, you have to clean the stage of the microscope with a clean piece of cotton, right? When you carry, there's a certain way that you have to carry. You have to use both hands because it's a heavy object. One hand should be used to hold the arm and the other hand should be placed under the base. Okay, and you have to tilt it towards your body when you take it from one place to another place. So likewise, okay, the microscope should be stored in an upward position. If the microscope is not used for a long time, the lenses should be removed and stored in a desiccator with silica gel. Don't change the lenses of one microscope to another, right? We will move on to the next. Question number 7. A diagram of a compound light microscope is given below. Label the parts of it. I am sure you all have done this correctly, okay? Because we did this many times now. What is this? This is the body tube. And these are objectives, objective lenses. This is the diaphragm. This is the mirror, eyepiece, cause adjustment knob, fine adjustment knob. This is the arm, stage clips, this is the stage, and the base. Right, children? Eyepiece, coarse adjustment knob and the fine adjustment knob. This is the arm, stage clips, stage, base, mirror, diaphragm, objective lenses and the body tube. Okay? We'll move on to the next. Right. Exercise number 8. State whether true or false. The microscope should be used only in an air-conditioned room. This is not true. You can use it just anywhere, but we have to make sure to clean it after using it. Right? So this is false. We'll write F here. It is ideal to avoid moisture and dust during use. That's true. Clean slides and cover slips should be used. That is also correct. Right? When observing liquid specimens, the microscope must be tilted. No, you should not tilt it, then the liquid will spill. Right? So when you observe in liquid specimens, you should not tilt the microscope. Okay, this is false. The microscope should be stored in a horizontal position, not horizontal position, it should be in a vertical or upward position, right? The microscope should be stored in a horizontal position is false. Change in the lenses of one microscope to another is not a problem. No, it is a problem because if one microscope has fungus on it, it will spread to the other microscopes as well, right? So it's false. After use, the microscope should be adjusted to low power. That is correct, right? So the damage to the lenses will be minimized in that way. When dirty, the microscope should be washed under running water. No, you should not. You should keep it away from water, right? That's also false. Right? The microscope should be used only in an air-conditioned room. It's false. It is ideal to avoid moisture and dust during use. That's true. Clean slides and cover slips should be used. That's also true. When observing liquid specimens, the microscope must be tilted. No, you should not. The microscope should be stored in a horizontal position. False, it's wrong. Changing the lenses of one microscope to another is not a problem. No, it is a problem. This is not correct. After use, the microscope should be adjusted to low power. That is true. When dirty, the microscope should be washed under running water. No, you have to keep it away from water. That's also false. Right, children? This was very easy. We'll move on to the next. Right. Number 9. What is the maximum magnification power of the electron microscope? Do you remember that? That is 500,000 times. 500,000 times. Right? Next one. What is the maximum resolution of the electron microscope? Do you remember that? 0.0005 micrometers. This is 200,000 times than that of a healthy human eye. Right? What is the maximum magnification power of the electron microscope? 500,000 times. Maximum resolution is about 0.0005 micrometers. Okay? Right. Additional exercise 10. This is the last exercise of the lesson. Write 3 instances where the electron microscope is used. Because of the very high magnification power and the resolving power, it can be used to observe viruses, including viruses, all the other pathogenic microorganisms, right? And it is used in crime investigation to get information about crime investigations, right? And it is used to observe the internal structures of cells, right, children? What else? It is used in genetic researchers as well, right? We will write number 1. Write 3 instances to observe pathogenic microorganisms. Number 2. Crime investigation. To observe the internal parts of cells. Right? Write 3 instances where the electron microscope is used to observe pathogenic microorganisms, to get information on crime investigation, to observe the internal parts of cells. So we have completed the entire lesson. So we learned a lot about microscopes. Now we know there are 3 types of microscopes as we learn in the lesson: simple microscopes, compound microscopes, and here electron microscopes, right? And then we learned how to use the compound light microscope in the lab, and we answered a lot of questions as well. So I hope you have a better understanding on this lesson.
To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.